Gracias por su presencia hoy aquí en la Fundación Aureces. Yo soy Carlos Martínez y es un honor, primero, agradecer a la Fundación Aureces y, por supuesto, a su director, Raimundo Pérez Hernández, por haber dado la oportunidad de introducir hoy al profesor Takmak para que nos dé una disertación sobre sus conocimientos sobre el cáncer, de la que estoy seguro todos aprenderemos. No creo que el profesor Takmak requiera mucha presentación, pero en cualquier caso sí que voy a hacer unos remarks, unas notas para aquellos que no la hayan oído con anterioridad, tengan el background donde poder insertarlos. I really believe, as I say, Professor Takmak doesn't really need any introduction, but just to mention a few points that I would like to make. He's a Canadian medical researcher, he's a geneticist, oncologist, and biochemist. He's professor at the University of Toronto and senior at the Princess Margaret Cancer Centre in Toronto, with interest in cancer diagnosis and therapy, cancer mechanisms and models, data science, computational biology, and lately, neural signaling as influence on immune responses. After a period of virologist, he started his scientific career as a virologist, where he identified many, and among many other genes, the oncogene CREL, that turns out to be a member of the NF-κB. He became widely known for his discoveries of the T-cell receptor in 1983, pioneering work in the genetics of immunology, a work that was described as the holy grail of immunology. Later on, in 1995, Professor Mack published another landmark paper with the discovery of the function of the immune checkpoint inhibitor CTLA-4. The combination of these two findings, the T-cell receptor and the CTLA-4, opened the path for the modern immunotherapy, without any doubt, the ideal situation that we have today to treat cancer patients, especially two drugs such as checkpoint inhibitors and chimeric antigen receptor, the CAR T-cell, as a means for cancer treatment. He has developed and established knockout mouse technologies and used these approaches to generate mutant mice-like genes involved in T-cell activation, development, and cancer, which have allowed countless scientists around the world to do high-level research. His generosity and sharing all those models with the scientific community reflect the scientific commitment and the human quality of Professor Mack. Professor Mack is also a founder of iOS Pharmaceutical, whose lead compound, IDF, was approved by the FDA for acute myeloid leukemia in August 2017. And I think today he will tell a little bit more about this drug. A drug that is the first drug specifically target cancer metabolins to be used in cancer treatment. For the discoveries of Professor Mack, he is awarded with countless prizes, among which many others, and I will not mention all of them, but just a few. The Emil von Behring Prize, the Gardner Foundation International Award, the King Faisal Prize, the Sloan Prize for General Motors, the Paul Ehrlich and Ludwig Prize, the St. George Prize for Progress in Cancer Research, the Novartis Prize in Immunology, and before yesterday, he got the last one, and I would like to mention that particular one, the Pethler Foundation American Association for Cancer Treatment. He's a fellow of the Royal Society, the National Academy of Science of the United States, and the American Academy of Arts and Science, and received many more than a dozen honorary doctorales, including Karolinska Institute, Göttingen, Zurich, Hong Kong, and Rome. As he used to say, the oncogenic revolution shows that cancer cell genome is too varied and the number of oncogenes too numerous. Is thus necessary, and that's something he has applied in his scientific career. When everybody runs in one direction, he would like to look backwards and see there might be something there, and that is what he found. And I think today he will present new anti-cancer treatments and new initiatives based by combining the tactics by which cancers evade anti-tumor immune responses, by targeting metabolic approaches that tumor cells use to survive conditions that make the tumor cells different from normal cells, or by manipulating a cancer cell response to excessive DNA damage. And finally, by also exploring a neoploid that is a future specifically associated to tumor cells. Tag, thank you very much for coming to Madrid and giving this lecture. The floor is yours.
Thank you very much, Professor Carlos Martinez, for this kind introduction. And I also like to thank the foundation for hosting this event. The title of my talk is Future Anti-Cancer Drugs, Put the Cart Before the Horse. So you think that sounds wrong because you put the cart before the horse. But in the last 3 years, everything has been very confusing. And so the COVID has brought us to a state of mind that is not just unusual, but sometimes really crazy. Now, I come from Canada, and Canada had a rule in the first year that you cannot go out of your house unless you walk a dog. And if you go out without a dog, the fine is 1,000 euros. So here it is, a Canadian in Quebec walking, I guess, her husband. And the judge decided that instead of 1,000 euros, it would be 3,000 euros, 2,000 euros, because the husband is also a person. So that's how crazy we are. So let's get back to the situation. Since about 20 years ago, cancer has become the #1 killer of basically all the developed countries. And in men and women, 1 in 2 men and 1 in 3 women will have cancer. Luckily, the drugs, the treatment, the radiation, the surgery had been very good. But nonetheless, there is still about half of the diagnosed patients will die from the disease. Here is Spain, which is in the EU pretty good in terms of the incidence and pretty good in terms of the mortality. But still the numbers are very big and we have to work very hard. Now the conventional treatment, the pillars of cancer treatment is #1, surgery, which was about over 100 years ago. Radiation was also about 100 years ago. And drugs, the concept that we can deliver something into a body was introduced by Paul Ehrlich, who won the Nobel Prize in 1910. And then Goodman and Gilman developed or not developed the first chemotherapeutic drugs to treat cancer. How many of you know what that drug was? Doctors pour drugs of which they don't live. to cure disease of which they know less into patients of whom they know nothing. In 1943, December the 7th, in Bari, Italy, German planes went in and bombed Bari because the Allied forces, the American, the British, the French, were already in the Bari area. The Germans went in and bombed. Immediately, there were many people who died on land and on the ships. But something happened. 10 days later, thousands of people died. 10 days later. What happened? An American medical officer, Stuart Alexander, and many smelled garlic. They said, could it be nitrogen mustard, which is, of course, banned for the use. But then why did these people die in the first 3 days? Then nobody died, and then 10 days later, thousands of people died. Stuart Alexander noticed that the lymph nodes of the patients who died were very small. So Gilman and Goodman said, I have cancer patients with very large lymph nodes. They call lymphomas. If nitrogen mustard actually can shrink, then why am I not treating my patients with nitrogen mustard? And they did. And it worked. And that was the beginning of chemotherapy. Now, as a footnote, here it is. At 7:35 p.m., thousands of people died. And this is the Great War secret. But that study of nitrogen mustard what then became the first chemotherapeutic drug. I'm not going to tell you where did the nitrogen mustard come from. The Germans said they did not use nitrogen mustard, but I'll leave that up to you. So, nitrogen mustard, first chemotherapeutic agent, 6-mercaptopurine from MD Anderson Hospital, methotrexate, Stanford University, etc., etc., etc. But you notice that by the turn of the last century, there are no more new drugs. Why? Because it's just too toxic. But it works. So we still use them today, and many people are still cured, but many are not. Then, in 1976, a paper came out in the journal Nature that Varmus and Bishop discovered that the virus that caused cancer in the chicken, called SARC, actually is not a virus gene. It is a chicken gene. In other words, the virus stole a gene from the chicken, changed it so that it works better and faster and then it caused cancer. Then that, of course, is the horse that pulled the cart. So if the horse can pull the cart, these are called oncogenes. They've been mutated or they've been amplified so that the car drives faster. Where the horse can pull the cart, stop the cancer, shoot the horses. And so you see all the drugs from 1998 all the way down to 2021, these are all oncogenes. And these are all the drugs that have already been approved to improve, extend the lives of cancer patients. In some cases, cure. But now 1 million cancer patients' genome have been sequenced. There are no new ones. Everything that was known was known. And there are millions of cancers that have been decoded. So Bob Weinberg, a very prominent MIT biologist, said, there are more paths to developing tumors than there are stars in the sky. Leukemia that have been very well treated has a single star. You can shoot that star. You can shoot that horse. Pancreatic cancer, one of the worst cancers. They have only one cut, one horse. But many are horses going backwards. They are called tumor suppressor genes. So it's a very difficult cancer to treat. And lung, breast, colon, it's a mixture that we are all trying to do. So, where is the future? No new oncogenes, no new drugs. The fire and the water, the physiology and the pathophysiology, the normal physiology and the good servants. Pathophysiology are bad servants because they tip the balance of your physiological functions. So you can rehabilitate it by intervening it and make it balance. Or when you can't do that anymore, you change the balance and kill the cancer cells. What are the 3 parameters? The first is metabolism. The second is DNA damage. And the third is immune activation. As late or early as 1966, Otto Warburg, a Nobel laureate, got together with several Nobel laureates in Lake Constance in Germany. I was just there. Actually, a couple of people in the audience were there last week. And they came to the conclusion, at that time, 1966, nobody knew what caused cancer. And they said, cancer is not caused by virus cancer. Hepatitis B virus causes cancer. Hepatitis C virus causes cancer. Human papilloma virus causes cancer. Why do you say this is called cancer? Because at that time, they didn't know that. Not caused by genes. Excuse me, cancer is caused by genes, gain of function, oncogenes, loss of function, tumor suppressor gene. And their conclusion in 1966 was the cause of cancer is the replacement of the respiration of oxygen in normal body cells by a fermentation of sugar. What are you talking about? If you like biochemistry, maybe you have an idea. If you don't like biochemistry, this is completely opaque. But what he meant was that something happened in a cancer cell, that the metabolism has changed. What Louis Pasteur noticed in yeast was that in the presence of oxygen, you do not make lactate. But what Otto Warburg noticed, cancer cells, even in the presence of oxygen, it makes lactate. Now, of course, that was almost 100 years ago. Knowledge was not somewhat that clear, but what really meant is that 2,700 genes in metabolism is somehow altered. But is it the cause or the effect? Here are the 2,700 genes. Nobody in this room knows anything, or all of them. Well, that doesn't stop us from being metabolic. This is, I found this on the web, glutathione. You can buy glutathione for €5 per bottle. You can take it. What does it do? Well, it protects you from oxidative free radical and cancer. It doesn't tell you it prevents cancer. Maybe it causes cancer. Well, it wouldn't make sense. But you know why I think they don't know? Because down here it says pregnancy. And then here, childbirth. It protects you from pregnancy and it gives you childbirth. Why? Because we don't understand metabolism. So, about 20 years ago, a group of scientists in our laboratory was studying Parkinson's disease. And there was a German doctor by the name Malte Peters, who was a gastroenterologist. And he was mixing the Parkinson's with a cancer gene. But by mixing them, they canceled out. So all of a sudden, we realized that cancer genes and metabolism is mixed. Maybe Warburg wasn't that wrong. At that time, many other scientists, Lou Cantley from Harvard, Craig Thompson from New York, other people were coming around, maybe metabolism and cancer has something to do. So to make a long story short, it is the mutations of the gain of function oncogenes or loss of function tumor suppressor genes. But they would kill the cancer cells. The cancer cells have to adapt. They have to change the metabolism so they can adapt. And that metabolism includes making more ATP, making new synthesis, and putting out the fire with the water, the redox. So 2 graduate students in my lab, a few years ago, Isaac Harris and Christian Chow, they're now both professors. one professor at Rochester University, Christine Chow, professor in Columbia University. They said if we tip the balance of an enzyme called glutamine cysteine ligase, what that does is it changes the amount of water, the redox, and upregulate the amount of reactive oxygen. Now, reactive oxygen, why? Reactive oxygen mutate DNA, it damages protein, it damages lipid, it's bad. So if you have more reactive oxygen, you should get more cancer. So the two of them put this mouse with no enzyme cysteine ligase, glutamine cysteine ligase, into 3 different cancer models. They were all expecting them to come down with cancer faster. It turned out it was the opposite. These mice did not get cancer. Well, not really. Their cancer was way delayed. You can see here, without glutamine cysteine ligase, more reactive oxygen, you have less cancer, whereas the normal mouse has a lot of cancer. So it turned out it is this adaptation when genetic change, it will kill the cancer cell. But a few of them get away with it, find a way, change the metabolism, and grass started growing again. And then another mutation, another mutation, another mutation. And eventually, it adjusted to all the metabolic changes. And these are all documented in papers from our laboratory. But I only want to tell you one story. This is a story which is, for those of you who know biochemistry, it's 100% biochemistry. The gene is called isocitrate dehydrogenase. It takes isocitrate to alpha-ketoglutarate in the mitochondria, and in the cytosol also takes isocitrate to alpha-ketoglutarate. What it turned out that gliomas, a terrible form of brain cancer, usually affecting young people, acute myeloblastic leukemia, about 20% of them, T-cell lymphoma that was discovered in our laboratory together with the Philippe Goulart in Paris, they all have mutation on a metabolic gene, isocitrate dehydrogenase. What does it do? It turned out it makes a metabolite that causes cancer. It's just crazy. You have a metabolite that causes cancer. In fact, there is this 2-hydroxyglutarate that needs to be degraded because it's a bad metabolite. And rare children will have an enzyme that cannot degrade this. And they all die before they're teenagers. Many die of brain tumors. So this is definitely an metabolite that causes cancer. So Japanese postdoc Sasaki, Christiane Knopp, a German neuropathologist, they worked out the mechanisms. It's called epigenetic changes, metabolic changes, and it causes some kind of a leukemia or leukemia, pre-leukemia, and use the mouse model for that. And this is another work that has just been submitted by a Japanese doctor in the lab to show the different combinations with other oncogenes and tumor suppression leads to different outcomes. And this is the work, just came out 2 weeks ago by a French postdoc, Julie Leaker, I was just in Marseille yesterday to celebrate her paper. She found that these cancer cells did not form on their own. In her case, she discovered that these metabolic changes, the changes in a T cell, but that T cell brings in B cells. So T cells and B cells get together to make antibody, but they interact for a few hours and they separate and they die, mostly the B cells. And they bring in a new one and they die. But this mutation forms a very strong bond between the T cells and the B cells. Instead of making antibodies, it makes cancer. And that's why this kind of shaking hand with the devil is very important. In other words, cancer cells don't just, I'm a cancer cell, I'm going to be a cancer here. All the people all the components, all the other cells in the same room, they together help you become cancer, or sometimes stop you from being cancer. And who is lucky, who's not lucky, that's it. Now, the good news. Myself, 2008, together with Luke Cantley and Craig Thompson, started a company to study metabolism in cancer. To make a long story short, 2 drugs. This is acute myeloblastic leukemia. 25% of the patients have this life extended. 25% have them extended a little bit more, and 50% of them about a few months. Well, it's good news, but it's also bad news because it doesn't do it for all the patients. Glioma, as I told you, is also got this metabolic mutation. This is a very young man, and you can see that after surgery, something is removed. But that if you take that drug over a period of about 2 years, the tumor has slowly shrank. Now, one patient is not going to be enough. So this paper just came out, and last night it was announced that it meets the endpoint. In other words, the drug from the very beginning went to FDA, Federal Drug Agency, and say, I expect so many patients to respond, and if I reach that, will you approve it? And to make a long story short, they met the endpoint. And soon we hope this drug will be approved for children and young adults' brain tumors. You can see here, by taking one or the other of the drug, at different doses, the tumor has shrank. This patient, 100%. This patient, 80%. This patient. It's not a cure. But when you're 30 years old with a family and children, to get 10 years is not a bad thing. That means we have to work harder. Now, interestingly, this mutation that we showed by another Japanese postdoc together with a Canadian discovered the mutation affect the ability to repair DNA. Each one of your cells has 2 billion base pairs. Every time it divides, it makes mistakes. Who can divide? And what kind of person can type 2 billion words without making mistakes? Does any one of you can do that? No. I make so many mistakes. My secretary said, stop typing, let me do it. It turned out this metabolic also make mistakes. And so genomic instability is when your DNA has many mistakes and it cannot repair properly. Children born with 3 chromosome 21 are called Down syndrome. They live, thanks to modern medicine, 30, 40, 50 years now. Children born with 3 chromosome 13 or 3 chromosome 18 die within 2 months of birth. These are cancer cells. Normal cells, 46 chromosomes. This cancer cell has over 100 chromosomes. It's not an exception. Breast cancer, renal cancer, ovarian cancer, pancreatic cancer, lymphomas. Look at them. It should be all here 48, 46. But look, this patient has 120. It's almost insane. How can they get away with it? That means they are unstable. They cannot make and repair the DNA. If that's the case, maybe if they can repair this much DNA, we will make them repair that much DNA. And what happened? They will die. So, we spent 5 years to look for extra genes during the cell division, and we found 2 of them. One is called polokinase 4, and that is called TTK. And they're involved in a cell cycle in the G1S checkpoint. What do? PLK4 is right here, this orange drop. When the cell divides, it goes 1 to 2, to 1 to 2, to 1 to 2. But in cancer cells, what are this? How many of them? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 1, 2, 3, 4, 5. Cancer cells are a lot more. This cancer cell is crazy. Look at them. 1, 2, 3, 4, 5, 6, 7. It doesn't even form what is called a centriole. The other enzyme is the green stuff. See, this is a normal cell. And a cancer cell, so many more because they have so many more chromosomes. These are the spindles. And look at this one. This cancer cell is totally abnormal. So if we target this enzyme and this enzyme, the green color and the orange color, what would happen? So we made drugs together with some really prominent Homer peers, made 24 drugs in 27 years in Eli Lilly. 2 of them were cancer drugs. They helped us. And here it is for this orange centrioles. Now, if they can get away with 5, 6, 7 centrioles, they cannot get away with 20, and they will die. And indeed, they will die. So this drug is now FDA fast track for leukemia. In other words, FDA said, I like it, It is not good enough yet. It's safe. It got some signals, but go and prove it to me that many, many more patients will respond. Second one, the spindles. Here it is. FDA fast-tracked this one 2 months ago. This is a breast cancer patient whose cancer went into the lung. This is an end-stage patient. The patient has gone through chemotherapy, palbociclib, and fulvestrant, and no more. And it goes here. So, interestingly, during dose escalation, in other words, you try many patients with many different and different combinations and different doses. We found 7 patients that responded to this drug, the spindle drug, and 5 of them are breast cancer. So we are quite excited about moving ahead. I'm going to end with immunotherapy, your immune system. Well, being Chinese, I'm allowed to quote Confucius. Confucius said, we all have 2 lives. The second one begins when we realize we only have one. This is a very, very sad statement. A year ago, I gave a talk in Idaho, in the US, and there were a lot of very, very prominent people there. And I gave this talk, and one of the prominent guests said, Professor Mack, where is the future of cancer treatment? I said, medical scientists have 2 lives. Second one begins when we realize immunology is the orchestra of all symphonies. Immunology is, of course, infection. COVID-19, terrible. Autoimmune disease, multiple sclerosis, lupus, rheumatoid arthritis, terrible. Transplantation rejection, terrible. Neurodegeneration, terrible. Cancer, terrible. So many immune cells. We can't even keep track of. Everybody studied a different. I studied this all my whole life. I studied this all my whole life. I studied this and this. Wow, you were brave. You studied both of them all my whole life. So it's very complicated. Another Chinese philosopher said, countless words count less than the silent balance between the yin and the yang. That's the immune system. It has inflammation because if bacteria is coming and it fire, the alarm go boo. Okay? And then the police come in and the detective come in. Which one started the fire? Which one is a bad one? Let me knock that one out so the whole organism, that is you, will survive. And then they build. T cells grows every 5 hours. Why? Because they have to catch up with the bacteria. If they lose that race, they're dead. Or you're dead. Or I'm dead. But when it's finished doing all that, it has to stop. You cannot let the police and the detectives take over the whole house, the whole building. But they do not really come back down to 0. And that inflammation is the cause of many, many diseases. Autoimmune disease, neurodegenerative disease, and cancer. So this T cell receptor, which is the one that we discovered, together with Mark Davis back in 1984. The T-cell receptor recognized a peptide, a little piece of a virus. It's just 8 or 9 amino acids, okay? And listed on something called HLA, which is a transplantation antigen. And T cells see both a flower and a pot. Both is like two codes. It has to be. And in doing it, it clears viruses, bacteria, parasites. It can even destroy cancer cells. Or can it? So this is a very famous picture by a very, very, very favorite painter, Wu Gunzhou. I wish I had met him, but he passed away about 10 years ago. And he called this painting Two Swallows. Which one of you can see the two swallows? So being a scientist, Simon Sinek, a British, lives in Hong Kong too. He said that most organization knows what to do. Few knows how to do it, but very, very few know why they're doing it. So that was a pure luck. This is me in 1984. This is Mark Davis. And we just, long time ago, were able to find the T-cell receptor. Because what, at that time, people said T-cell receptor is part of immunoglobulin, part of a banana, part of an apple. There was no rationale to it. We took an approach, and by accident and by luck, we found it. And we found the key to the car, the T-cell receptor. But every car has to have an accelerator and has to have a brake. Well, the brake was found by Pierre Goldstein in Marseille, but he didn't know it was a brake. So we knocked out the gene that was the brake, called CTLA4, make a mouse without that gene, and the mouse died in 2 weeks because these are all the T cells. So in other words, that gene is the brake. If you don't have the brake, it just keeps going and going and going. So there's the brake. Another brake is found by the Japanese called Tasuku Honjo. But I had no idea. We discovered it was a brake. And I was saying, I work in a cancer hospital, remember? And this is, you can't. I went to my boss and I said, can we use this? He said, no. Look at the mouse. If you shut down that brake, the mouse or the patient would die. And this is Jim Allison. He lit the candle while I cursed the darkness. And of course, he put an antibody on the brake, and boom, the fourth pillar of cancer treatment. It's simple. Millions of patients now been treated in the last 13 years, and many spectacular responses. But Not all of them. Some of them do better. These are overall responses. Not many, Some have about 50% response, some have 30% response, some have no response. But it's still a fantastically important concept, and the two of them won the Nobel Prize. Very, very well deserved. Okay, now what? How do you treat the rest of the patients? How do you make the ones that respond continue to respond? The whole world went crazy. 2,000 clinical trials are going on, studying checkpoint, co-stimulation, Tregs, DNA damage, metabolic, etc. You use microenvironment, or some drug companies say, hey, I have a drug that doesn't work. Let's combine with immunotherapy. Or I have a drug that's already selling for a lot of money. Why don't I combine immunotherapy with sell more? So it's a complete chaos. Unfortunately, so far, it hasn't worked. Very few combining with immune checkpoint has been approved. Why? Because the tumor microenvironment is totally crazy. It's very, very, very complicated. I can't even begin to study it. But sometimes the T cells be working for too long. They get exhausted. It's my granddaughter. How do you get, his name is Sadie. How do you get Sadie back up? Well, you add an enzyme, a cytokine called interleukin-7. This is an Australian postdoc in my lab, and Thomas Kelskaus, a Swiss postdoc in Pam Mohashi's lab. We discovered that interleukin-7 can wake these cells up. There is a mouse with cancer, and they die within 100 days. If you kick in the immune system, it works for another 10, 20 days. But if you add interleukin-7 on top, the mouse lives another 100 days. Another is the wall. Here, this is the tumor. This is the cancer. This is the cancer. Look at these killer T cells. They can't get in. They can't get in. I want to go in to kill them. I want to go in and kill them. I want to go in and kill them. I'm sorry, there is a wall here. And this wall says, nope, you cannot come in. It's almost like Helen of Troy. How do the Greeks go in? You use the Trojan horse. What is the Trojan horse? Now I'm going to end with another story of metabolism. There's something called choline. It's in a, if you really want to, you can buy choline in the drugstore, food store, or you can just eat a lot of broccoli, probably safer. Choline is involved in three things: epigenetic changes with methionine. It also is involved in making sphingomyelin, but choline is involved in neurotransmission. It helps your brain, your neurons, to send messages to each other. But no one would have expected this. 40 years ago, scientists were finding that if you leave choline and methionine out, the rats and the mouse will come down with liver cancer. So, if it's choline and methionine, it's epigenetic change. 10 years later, they say, hey, hold a minute. You don't need to leave out methionine. Just leave out choline. Then all of a sudden, it's not just epigenetic through DMT1. It could be sphingomyelin and God forbid, it's a neurotransmitter. So it's back. Any one of these can be possible, causing this liver cancer in 100% of all the rats and the mice. 170 years ago, there is a German called Rudolf Wagner. He stimulated the nerve of a dog, and the spleen moved. Now, this makes sense because spleens are immune cells. The spleen is only immune cells. Nothing there. But if you stimulate it, the immune cells are agitated. Now, his friend, Jakob Handy, did something I considered totally unacceptable today. He stimulated the splenic nerve of a decapitated human and saw the same results. Well, don't try it. Please don't try it at home. And then, of course, eventually people track the nerves. So there's the central nervous system, and there is then the sensory nerves, and this is called autonomic nerves. These autonomic nerves, these are nerves that will help your body function without you thinking about it, okay? Because you see a lion outside in the street, and you by yourself, immediately your sympathetic nerves will pump out epinephrine or adrenaline. Now you know that. Oh, I've got adrenaline. And your heart pumps boom, 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 boom. And then you got into a car, you're safe. And then the parasympathetic nerves come in and they make acetylcholine and it slows down. I'm safe. So the first one is fight and flight. The second one is rest and digest. This is norepinephrine. This is acetylcholine. So when Wagner and Handy stimulated the nerve, the spleen moved. And in 1929, they eventually found acetylcholine. Bingo! Everything works. Brandon and Rand said, if I stimulate the nerves, they make more acetylcholine. Bingo again. Everything is fine. But what nerves make acetylcholine? Parasympathetic nerves. But only sympathetic nerves are found in the spleen and the lymph nodes. So they should be making norepinephrine. Then who makes acetylcholine? The nerves are actually sympathetic nerves, norepinephrine, but you find acetylcholine. So science progressed, not because scientists changed their minds, but because scientists attached to erroneous will die, and they're replaced. 50 years went by, no solutions. And then comes Kevin Tracy, a neurosurgeon in New York. I had dinner with him about 15 years ago in the Metropolitan Club. He was just sitting next to me. He introduced me. My name is Kevin Tracy. I'm the neurosurgeon. I said, oh, yeah, that's good. Yeah. I said, this is going to be so boring. And I said, what do you do? He said, yeah, well, I do neurosurgery. I said, you do any science? He said, yeah. So he told me all this story about how acetylcholine and norepinephrine is all mixed up. I said, hey. If you make a mouse that the enzyme that makes acetylcholine, called acetylcholine transferase, chat, is linked to a light bulb, a green color. then you know who's making acetylcholine, right? So together, we discovered the immune cells are making acetylcholine. That is why the T cells and the B cells. And so, make a long story short, an infection, inflammation sends the brain. Now the brain sends back norepinephrine. Norepinephrine stimulate the T cells together with the T cell receptor to make acetylcholine. Why? What do you want to need it for? But it's not really that impressive, isn't it? 2% of the mouse T cells make acetylcholine, 0 CD8 T cells, 2% CD4 T cells. This is an American postdoc from the South, Alabama, and I went there to give a talk. Students invited me, and I say, hey, let's come to Toronto. I've got this puzzle that you need to solve. What are these 2% of cells doing? I said, no, it's too cold. So, okay, well, try it for 3 years. So she came. So she took that mouse that whenever they make acetylcholine, the light bulb turns on. It's called a GFP. And she infected the mouse. 8 days later, 1/3 of the cells are pumping out acetylcholine. These are immune cells. They're pumping out neurotransmitter. What's going on? CD8 T cells, 0. And then after a virus infection, another 30%. In fact, all the immune cells at the peak of the infection, half of them pump out acetylcholine. Why would immune cells need a neurotransmitter? So we sent in the paper, it was rejected, because this is just correlation. Okay, so Maureen, can you stay 2 more years? I said, no more. It's too cold here. We have to go back. My parents in Southern California, they said no. I said, well, but don't you have a boyfriend? I said, yeah. Canadian, right? Yeah. So actually they got married. So I have 2 more years. And now, this is the experiment. The experiment, if you delete in T cells the ability to turn on the light bulb. Now remember, you cannot turn on the stuff acetylcholine in the whole body because your nerves, every one of you sitting there, acetylcholine firing billions by second now. Okay? If you delete that, so here you have a mouse with T cells cannot make acetylcholine. And what happens? When T cells cannot make acetylcholine, when they can make acetylcholine, it's the blue dots. They clear the virus in 30 days. If you cannot have acetylcholine in your T cells, you can't clear the virus, and the mouse dies. So a neurotransmitter is helping your immune cells to fight it. Many functions I can't get to. I have to end because it's getting late. But one of it is to help dilation. So when I was young in the University of Wisconsin, my friends say, hey, Tak, BYOB, Friday night. So bring your own bottle of BYOB. T cells bring their own acetylcholine to open the door so they can get out. I can't go into it. Half my labs now involve many things are going on that the immune system uses acetylcholine. Together with a Swedish scientist, we showed it affects blood pressure. Acetylcholine from your immune cells. German postdoc found that if you have acetylcholine in your T cells after experimental autoimmune encephalomyelitis, which is the MS, multiple sclerosis model, you get the disease and the mouse die. If you don't have it, you don't get the disease and the mouse doesn't die. A Chinese postdoc showed that in liver cancer, if you don't have neurotransmitter from your T cells, you have more tumors, bigger liver, and die faster. So this is all just animal stuff. So, all right. So I work in a hospital. I'm on the 8th floor, and then 1, 2, 3, 4, 5 floors are all doctors, and then 11, 12, 13, 14, 15, 16, 17, 18, they're all patients, right? I go to drinks with these doctors and say, hey, Jeff, Dr. Watson, Eric, Dr. Chan said, can you do something for me? I said, yeah, what do you want? Can you measure the choline level, acetylcholine level, in the blood of your patients undergoing immunotherapy? Why? I have no idea. So after 2 years, they did it. And they came to me. And it shows that if you have high choline and acetylcholine, you live. If you have low, sorry. I have no idea what's going on. But there's more evidence. If you have more receptors, you live longer. These are acetylcholine receptors. So I'm going to end with just 4 more slides. Go back to the beginning. The T cell receptor, which we so luckily discovered back in 1984, recognize part of a virus and part of your own self. CAR T is the brilliant work of Carl June, Michel Stellerline, and an Israeli professor. They took the T cell receptor and they attach an antibody on the outside. This is like Jason Bourne. You know, you've seen Jason Bourne, right? Jason Bourne would fly in a helicopter, jump in the middle of Moscow, kill everybody, goes into the KGB headquarters, kill the director, and jump out of the window 20 floors, and he's after Putin. I hope he wins. I may get into trouble. And this works. Look at this. It's now been approved for leukemia, lymphoma. Patients 10 years ago, terminal cancer, some of them are still alive, by just sharpshooting the direct target. And this is the T cell receptor. It's not a target, it's a peptide in HLA. And in order to do that, together with Professor Naoto Hirano, we have been doing a lot of maneuvering to make sure we have enough T cell receptor for everybody in this room and find the bad targets on the cancer cells. So these are my 2 mentors, both have since passed away. Ernest McCullough discovered hematopoietic stem cells. Howard Temin discovered reverse transcriptase, received the Nobel Prize. Simply sail in a new direction, and you could enlarge the world. I'm partly here because I came for a European reunion of the people in my lab. There's been 150 from all parts of the world. 6 are from Spain, and one of them is in the audience. And the one in the audience is this one. Professor De La Pompa, can you stand up? Thank you very much. He's a brilliant guy. I'm so very lucky. This was this reunion we just had, and these are the people over the years that I've talked about the work. Thank you very much.